Good morning, good afternoon, good evening, wherever you are. And welcome to this George Macdonald Medal talk, which will be given by Professor Arjen Dondorp, who was awarded the medal in 2023. And we're also celebrating for next week, a little bit early, World Malaria Day. So welcome everyone to this wonderful medal talk. Now, for those of you that I haven't met yet, I'm Tamar Ghosh. I'm the Chief Executive here at RSTMH and delighted to be introducing the talk for you. For those of you that don't know RSTMH, we are a charity, a membership society for those working or interested in tropical medicine and global health. We have big ambitions around eradicating, eliminating disease and improving health equity. We're based in the UK and we carry out lots of different activities, including giving out medals and awards like the George Macdonald Medal to celebrate success in our sector. We also give out early career grants for those who've not had research funding before. This year has a deadline of Monday, so very soon. And we also have 2 scientific journals that we publish monthly and every 2 months. And you can read about all of these on the website. And a bit like today, we have events that happen in person and virtually across the world in all topics that you can imagine within the fields of tropical medicine and global health. So a bit of housekeeping for today's medal talk. Please, as we go through and hear from Professor Arjen Dondorp, do please think about questions you might like to ask him and post them in the Q&A function, which is at the bottom of your screen. There should be time for questions at the end of his talk. All right, so now I have the pleasure to introduce the chair for the talk, Professor Olugbenga Mokuolo, who nominated Professor Arjen Dondorp for the medal, and he will introduce him. Now, just to say, the bios for both of our incredible speakers today are very long. They will appear in the chat so you can read them. Welcome, Professor Olugbenga.
Thank you very much, Tama. It gives me great pleasure to be here. I want to welcome everyone here once again. And I will shortly be introducing Professor Dondorp. Let me share his introduction with us. Yeah, so Professor Dondorp is delivering the George Macdonald Medal talk today. And Professor Arjen Dondorp is an infectious disease and intensive care physician based at Mahidol, Oxford Tropical Medicine Research Unit, popularly called MORU, in Bangkok, Thailand, where he's the Deputy Director and heads the Malaria and Critical Illness Research Department. He is a professor of tropical medicine at the University of Oxford, professor of Translational Global Health at the University of Amsterdam. He's a visiting professor of clinical tropical medicine at Mahidol University in Thailand, and is also fellow at the Institute of Global Health and Development and a fellow of the Academy of Medical Sciences in the UK. His main research interests include pathophysiology and treatment of severe malaria, antimalarial drug resistance, new treatments for drug-resistant falciparum malaria, and improvement of intensive care practices in developing countries. Regarding his citation, I had collaborated and related closely with Professor Dondorp over about 18 years now. Over these years, I have come to appreciate that he is an astute clinician and an excellent researcher. He thinks through practically every line in a process and hardly does he skip any details in his scientific elucidation of facts. And I'm really amazed at his capacity to relate with so much information at the same time. From the pathophysiology of malaria through malaria chemotherapy to artemisinin resistance, Professor Dondorp has been phenomenal, leading studies that has shaped our understanding and treatment of malaria globally. Over these years, I have come to appreciate that he's an excellent researcher, and therefore I could not think of any other person to nominate for the George Macdonald Medal Award when the opportunity came up. So it was a no-brainer, really, nominating Professor Arjen Dondorp. And receiving the news of his selection for the award was a career high point for me. I am proud personally to have been associated with his award, and it gives me great pleasure today, distinguished ladies and gentlemen from all over the globe, wherever you are joining this meeting, to present to us Professor Arjen Dondorp to deliver this year's George Macdonald Medal Award talk. I wish you a very interesting session and great time of elucidation around his works. Thank you. You're all welcome.
Thank you very much, Professor Benga, for this very nice introduction. And of course, I'm very honored both that I've received the medal and that I can give the presentation today. I will start sharing my screen. Maybe you can confirm that you can see it.
We can.
And the problem with these online lectures is that you have no idea who the audience is. So it will be about antimalarial drug resistance in falciparum malaria. It's a very hot topic because the problem is increasing at the moment with artemisinin partial resistance emerging now independently in Africa. So I will focus on falciparum malaria, but you probably all know there are several Plasmodium species that can cause disease in human beings. You see them in this slide: Plasmodium falciparum, Plasmodium vivax, Plasmodium malaria, Plasmodium ovale, and then there's the monkey malaria, Plasmodium nosai, in long-tail and short-tail macaque monkeys that can easily jump species. But falciparum malaria is the big killer. 95% of all malaria deaths are due to Plasmodium falciparum severe malaria. So I will focus on that today. And the other basic fact you all know is that the Plasmodium parasite is transmitted by the female Anopheles mosquito. More than 40 important species that can transmit the disease, different between the continents. The Anopheles mosquito injects the sporozoites into the body, and then the parasite quickly goes to the liver and then sits there for almost a week, and then it multiplies from 10 parasites to 10 to the 5th parasites. Then they are released in the circulation, where they live in the erythrocyte, and they have a 48-hour life cycle where they develop from a small ring to a trophozoite to a Scheißeln stage when they start to divide. And then every 48 hours, you get an amplification in the number of parasites, around 10-fold every 48 hours. And you get feverish when you have around 10 to the 8th parasites, and you get severely ill when you have 10 to the 12th parasites. And then after a few cycles, part of the parasites will develop into gametocytes, the sexual stage of the parasite that can be taken up again by another Anopheles mosquito, where the sexual part of the cycle starts, and then the cycle is round. Malaria is a very different disease in sub-Saharan Africa compared to Asia, where I live and work, because the transmission intensity is so much higher in many parts of sub-Saharan Africa. where a child is bitten almost every day by an infected Anopheles mosquito, and so continuously exposed to the parasites. And because of that, the children slowly develop partial immunity called premunition, which more and more protects the individual. So that severe disease is almost entirely only happening in children below the age of 5. And all the children and adults no longer get severe disease. They can still get a fever, or they can be asymptomatic carriers that can still transmit the disease, but don't cause illness in those individuals. Compared to Asia, where the transmission is much lower, and on average, someone gets infected once every year, and that makes that this premunition is not really establishing itself, and all ages can be affected by the parasite. And because of behavior, the young adults are most exposed to the infection. And you see that in the epidemiology of the disease. So very different situation there. That slow transmission of the parasite makes that it is more vulnerable to the development of drug resistance. And that has several causes. In a high transmission setting, the individuals, the children, are infected, but also the adults, are infected by multiple strains of the parasite. That makes that when one parasite strain is resistant, for instance, because of a genetic makeup with point mutations, that genetic makeup is easily lost when the infection is transmitted to the mosquito, because the mosquito will also take up different strains of the parasite. And then during crossover and recombination during meiosis in the mosquito, it will lose this genetic makeup and the resistance pattern. In addition, the drug pressure will almost always be lower in these high transmission settings, because you have a big burden of parasites in asymptomatic carriers that do not seek treatment and don't take antimalarial drugs. So you have less drug pressure in that situation. And overall drug pressure is lower because there is less use of antimalarial drugs in these very high transmission settings. And then there's immunity. So in the older children and adults, the immunity helps to clear the infection. So if you have just a little bit of resistance to an antimalarial drug, the individual can still quite rapidly clear the infection, so that transmission of those resistant parasites is stopped by that. And then finally, I put there is a barrier against intercontinental spread because of barriers in Anopheles species. Nevertheless, this is not an absolute barrier to antimalarial drug resistance in high transmission areas. And we know that very well from the past. Chloroquine resistance started here in the Greater Mekong subregion already in the late 50s, and then slowly spread westwards to India, then crossed the ocean to Eastern Africa and spread from there. And there it has caused big harm, and I have a next slide on that to illustrate that. After chloroquine became unusable in the Great Mekong subregion, it was replaced with sulfadoxine pyrimethamine. Well, pyrimethamine resistance also rapidly evolved in the Great Mekong subregion, and then also followed the same route westwards and then crossed the ocean towards Africa. So it did arrive in Africa, and there, especially chloroquine resistance has caused many, many deaths. And that was not early on recognized. And even nowadays, there are relatively few scientific publications documented that. I put on an example here on the slides, which is a study from verbal autopsies. You can criticize that, but it does show that in the 90s, when overall childhood mortality was going down in Africa, malaria attributable mortality was clearly going up in Eastern Africa when chloroquine resistance was spreading there very quickly, and the children were still being treated with chloroquine. So just to say, it's not an absolute barrier, and once antimalarial drug resistance well established and the children are still treated with the same antimalarials, it can have devastating effects with an increase in malaria and an increase in malaria deaths. Here in the Greater Mekong sub-region, where drug-resistant malaria is a much bigger problem historically, for the reason I just outlined, in the 90s, it was a very dire situation with all the different drugs losing their efficacy. As you see here on the left side, so chloroquine was lost, sulfadoxine pyrimethamine no longer useful, mefloquine, used as a monotherapy those days, quickly lost its potency, first at a lower dose, later at a higher dose. And we were really saved by the artemisinin drugs. So the artemisinin compounds discovered in China, 2UU awarded the Nobel Prize for that, are derived from Artemisia annua, a plant you see there on the picture. It's an endoperoxide. It has very rapid action, very potent, and has a broad asexual stage specificity. So all the other known antimalarial drugs that we currently use only kill trophozoite and Schizon state parasites, but the artemisinins already kill the ring stage parasites. So broad stage specificity, very potent, and also gametocytocidal, early stage gametocytes, so with that also reducing transmission. And it took a while before they were widely adopted, but nowadays they are first-line therapy in all malaria endemic countries. Here you see the 5 main artemisinin-based combination therapies that are currently being deployed. Artemethyl-lumefantrine is most widely used, artesunate mefloquine, DHA piperacin, artesunate amlodipine, and artesunate pyroneridine, the newest ACT that is being marketed. So we thought for a while that we would be safe with these artemisinin-based combination therapies because the artemisinins are so potent and they have a very short plasma half-life with a half-life of around 1 hour, which means that the parasite, we thought, has very little time to develop resistance. Nevertheless, around 15 years ago, we noticed that here in the Greater Mekong subregion, in Western Cambodia to be precise, clearance of the Plasmodium falciparum parasite became much slower after a patient was treated either with artemisinin monotherapy as a start or with artemisinin combination therapies. So instead of around 10 to the 4th decline in a 48-hour life cycle, only 100-fold decrease in parasitemia was achieved after 48 hours. At that moment, the ACTs themselves were still quite effective, but nevertheless, we thought that this loss in potency was a worrying sign. The next step was that it was discovered what the molecular mechanism for artemisinin resistance was, and mutations in the so-called CALC gene, the propeller region of this CALC gene, appear to be very closely associated with this slow parasite clearance phenotype in artemisinin-resistant parasites. In the beginning, there were many different CALC mutations being identified. More than 70, in fact, have been seen. The parasite only allows one CALC mutant. If there are several SNPs in the gene, then apparently the parasite is no longer viable. But in the beginning, there was this wide range of potential CALC mutations, but only a few of those appear to be successful, as I will show you later. Another thing that was discovered in laboratory studies is that resistance of the Plasmodium falciparum parasite was mainly restricted to the ring stage of the asexual parasite in the erythrocyte. And that you can study that quite nicely in vitro with the ring stage assay, where you give a pulse of dihydroartemisinin to a culture with Plasmodium falciparum in the ring stage, in the early ring stage. And that is where you see this big difference in survival of the parasites between CALC mutant and CALC wild type parasites. If you let the parasite mature to the trophozoite stage, then there is not a difference between CALC mutant and CALC wild type parasites. By the way, the mature Schizon stage parasites, also there, the artemisinin resistant parasites are less affected by the drug. So that is summarized in the cartoon on the right side, that the broad stage specificity of the artemisinins from the ring stage trophozoite to Schizon stage is reduced to only the more mature stage in artemisinin resistance. And that is the reason why artemisinin resistance is also called artemisinin partial resistance, because it's not the whole life cycle that is affected, but it's mainly this ring stage that is affected by artemisinin resistance and the very mature Schizon stage. The pathobiology of artemisinin resistance, there I could talk a whole hour about that. There are many different ways that mutations in the CALC protein can contribute to artemisinin resistance. It's nicely summarized in a recent review here in this cartoon. One of the things that are affected is hemoglobin uptake by the parasite. Hemoglobin is the food of the parasite. It's taken up through these cytostomes, and the CALC protein co-localizes with proteins in the cytostome that are needed for the uptake of hemoglobin. And then hemoglobin uptake is reduced. Hemoglobin is needed for activation of artemisinin. Artemisinins have this endoperoxide bridge that needs to be activated by the iron 2+ in the hemoglobin. Well, if there's less hemoglobin available in the food vacuole of the parasite, there will be less artemisinin activation, and there's also less proteins available for the parasite to grow. So one of the consequences of it is that you see arrested development, so slowing down of the ring stage development. And you can see that if you follow closely the parasite age in artemisinin-resistant parasites compared to wild-type parasites. This was, by the way, one of the reasons why we thought that we might overcome the problem of artemisinin resistance by increasing the dosing frequency of the artemisinin combination therapies from once daily for 3 days, what the normal regimen is, to a twice daily dose. However, if we tested that in the field, and this is a study that is now more than 10 years old, we did not see that importantly improved the efficacy of the treatment. And we were a bit puzzled by that. We think it's because it's not only slowing down of the development of the ring stage parasites, but the parasites also becomes refractory to the effect of artemisinin for around 2 to 3 hours. And if you combine those two effects and you put that in a model, then you can explain the clearance curves of the split dose therapy compared to the single dose therapy. And then there are other mechanisms conferring artemisinin resistance. There's an upgraded and upregulated unfolded protein response in artemisinin resistance parasites and several other processes going on. So what does it matter for the individual patient that he is infected with artemisinin partial resistant parasites? Because the artemisinin still have an effect, although it is much slower. The ACTs, they can still cure the patient as long as you have a fully effective partner drug. And that we saw at the beginning of the problem here in the Greater Mekong subregion. You had artemisinin resistance, but as long as it was combined with a fully effective partner drug, you still saw very high efficacy of the artemisinin-based combination therapy. However, if you see a small decline in efficacy of the partner drug, then you see ACT failure. And that we have witnessed on a big scale here in the Greater Mekong subregion. So with the failure of, with resistance emerging in mefloquine sensitivity, in piperacin sensitivity, and now more recently with artemether-lumefantrine. So both of the partner drugs, both components of the ACT contribute to efficacy of the combination. If you have only artemisinin resistance, you do see a small decline often in the efficacy of the combination, as nicely illustrated on the Thai-Myanmar border, where artesunate mefloquine was introduced at a moment that there was already quite widespread mefloquine resistance. First, the combination was still highly effective, but as soon as mefloquine resistance appeared, the combination failed. Here you can see more than 40% treatment failure after 42 days. Whereas if there was no mefloquine, no higher grade mefloquine resistance, for which we have a marker, MDR1, multiple copy numbers, then we saw just a little bit of failure of the combination. But there's another effect why artemisinin resistance is so dangerous, and that it facilitates the development of partner drug resistance. So we have seen that with mefloquine very clearly on the Thai-Burmese border, where artesunate mefloquine was a very good combination, but as soon as artemisinin resistance appears, as you can see here in those bars, the Celts mutant are the black bars, then quickly mefloquine resistance very rapidly increased and made the combination failure so high. And we think that that is because of the reduced efficacy of the artemisinins after 3-day therapy. Instead of a 10 to the 5th or 10 to the 6th decline in parasitemia, you only have a 1,000-time decline. So many more parasites than see only the partner drug, because the artemisinin has a very short half-life. The partner drug is usually less potent, but has a much longer half-life and is there to mop up the remaining parasites. So we have seen that effect, that artemisinin resistance facilitates partner drug resistance very clearly with artesunate mefloquine, but also with DHA piperacin. And once you have failure of the ACT, then you have very rapid spread of this problem of the resistant phenotype. And that has several reasons. One, you don't cure your patients anymore, so then they can transmit the disease on. But in addition, these recrudescent infections, they always carry more gametocytes, so more sexual forms of the parasites. And that is a known effect for drug resistance in general. It was first observed with sulfadoxine pyrimethamine, where you had much higher gametocyte rates when resistance to SP emerged. The quality was a bit less, but still overall it had a big effect on transmission, increased transmission specifically of the resistant parasites. But the same effect has been shown for mefloquine, for chloroquine, and most recently also for the artemisinins, where a recrudescent infection after an ACT has around 9 times more gametocytes than the initial infection. And of course, that then helps a lot in spread of the resistant parasites. In addition, the artemisinin-resistant parasites also spread without drug pressure from the artemisinins, because it appears that artemisinin-resistant parasites, they transform earlier to gametocytes. There's an upregulation of this AP2G gene, which is an important gene for committing the parasites into gametocytes, which has been shown in vitro. It also explains the epidemiology. For instance, in Myanmar, there's this CALT mutant, the 446 mutation, that only gives a little bit slower parasite clearance, but nevertheless it was selected for and spread rapidly in Myanmar, which you can only explain if it has higher transmissibility. In addition to that, in artemisinin-resistant infections, the gametocytes, so usually the younger stage gametocytes, are killed by the artemisinins. In artemisinin-resistant infections, we know, especially for the male gametocytes, that they have become less sensitive to the artemisinins. So another reason why these infections are easier spreading and can establish themselves. So all this happened first in the Greater Mekong subregion, where first we had all these different CALT mutations circulating and next to each other. But a few of those CALT mutations were the most successful, and here in the eastern part of the Greater Mekong subregion, we had this CALT 580Y mutation, and that was really driven by the widespread use of DHA piperacin, and we saw selection of this single lineage of 580Y parasites spreading all over Cambodia, and very resistant to both DHA and piperacin. So it's also picked up CRT mutations that were conferring piperacin resistance and plasmapsin 2-3 amplification. Then the first-line therapy was changed away from DHA piperacin back to artesunate mefloquine, and then we saw a change in the Celts mutant that was most successful, away from the 580Y. So nowadays we see other Celts mutants coming up again. So this was all happening over the last 15 years in the Greater Mekong subregion, where it started with artemisinin resistance, which facilitated the development of ACT partner drug resistance, making the ACTs failing on quite a large scale, with 4 of the ACTs no longer working in Cambodia, Thailand and Laos also in a poor shape, Vietnam closely followed by Vietnam. So this was an emergency that was well recognized. What about severe malaria? Well, at the same time, the transmission of falciparum malaria had gone down dramatically in the Great Mekong subregion. So we see and we saw little severe malaria, and we don't have big series whether the efficacy of injectable artesunate, which, as you know, is the first-line treatment for severe falciparum malaria, is also jeopardized. So on the left side, you see how much better artesunate is compared to quinine in saving lives in both children in Africa and adults in Asia compared to quinine. We only have these case series showing that, yeah, probably we should be worried in artemisinin-resistant severe malaria that artesunate is no longer always a very effective treatment. Here you see a few cases where the parasitemia did not go rapidly down as we would expect after injectable artesunate, and a few of those patients died. Now that we have artemisinin resistance emerging in Eastern Africa, we are of course very worried whether injectable artesunate, intramuscular or intravenous, is still as effective for the treatment of children with severe malaria. And there are few alternatives only. So if we go back to quinine, we know that there is 25% higher mortality. There is a new drug being developed by Novartis, cipargamin, which needs to be trialed in these patients and compared to artesunate in children with severe malaria and artemisinin resistance. So what happened over the last 50 years in the GMS after artemisinin resistance was clearly recognized as an important problem, and the ACTs were losing their efficacy in treating patients? Well, it was recognized that if you want to get rid of that problem, you have to eliminate all falciparum malaria, because there are no drugs available, especially at that time, that did not contain an artemisinin component. So you needed to eliminate all malaria to get rid of the problem. And several plans were being written. There was high political commitment and backup for those plans, and there was a lot of money available from the Global Fund, who started this initiative, the Regional Artemisinin Resistance Initiative, since 2014, where they fund a regional approach to malaria elimination with a multi-stakeholder organization of it. So close collaboration between NMCPs, with the civil society organization, with academics, with the WHO, all working together, making plans together, and that has established a network of village malaria workers in the 5 countries of the Greater Mekong subregion for early diagnosis and treatment, for surveillance, for distribution of bed nets, and for elimination interventions, including targeted mass drug administration, individual follow-up of and active case detection after individual infections. So this has been going on since 2014 and has been highly successful, at least in the eastern part of the GMS, where really falciparum malaria has now almost disappeared, a few hundred cases per year only. And you see that here in these maps, which come from the WHO database. All the countries, they have strong surveillance now in place, which are shared between the countries, and then the WHO makes these nice maps. And you can see from left to right that the red zones are fading away more and more. However, in Myanmar, we have a huge problem where the health infrastructure has crumbled after the military coup now more than 2 years ago. And that is a real problem, and malaria is coming back there, unfortunately. So the Eastern GMS, very successful in their malaria elimination program. But then in Africa, where we were afraid that the problem would spread from the Greater Mekong subregion to Africa, that was one of the reasons to try to suppress transmission as good as we could here in the region. But then there has been independent emergence of artemisinin resistance in East African countries. First in Rwanda, where the 561 mutation, Kelch mutation, has now been clearly established, and later also in Uganda, especially in northern Uganda, where the 675 and the 469 Kelch mutations are now present in up to 30% of the infections. So Uganda is, in Rwanda, you could say that was quite low transmission at the moment that it appeared. They had a quite successful malaria program. Northern Uganda, however, is a high transmission setting. So it was surprising that also there you had an independent emergence of artemisinin resistance. People say maybe it was because it was after a very stringent implementation of malaria control with bed nets and early diagnosis and treatment that was then relaxed after a while. That was the bottleneck that facilitated the emergence of artemisinin resistance in those countries. Nevertheless, Uganda is a real worry, where now you have a combination of artemisinin resistance and also high transmission. So, for instance, also severe malaria is an important problem in those northern Ugandan provinces. And as I said before, we are worried that IV artesunate might be less effective in saving lives in children with severe malaria there. This slide is to show that we know for sure that it is independent emergence of artemisinin partial resistance in those countries. And you can look at that by looking at the flanking regions of the Kelch SNPs. The flanking regions have a very different genetic makeup than the parasites from the Greater Mekong subregion. So it's definitely not spread from this part of the world to Africa. It has emerged independently. And the problem is getting worse and worse. Here, a few recent publications listed, and on the right side, a recent map in a review article by Phil Rosenthal, where you see those different mutations. At the moment, it is still those different mutations in the different locations, the 561 in Rwanda, 675 and 469 in Uganda, and the 622 in Eritrea and Ethiopia. It has been recognized as something very serious. So the WHO has developed this strategy to respond to antimalarial drug resistance in Africa after it became clear that in these East African countries, artemisinin resistance has emerged. It's quite a generic document with multiple possible interventions which now need to be translated into country-specific plans to target this problem. And why should we worry about artemisinin resistance in these African countries? I already mentioned severe malaria, but of course we also have to worry about the efficacy of the artemisinin-based combination therapies because they are first-line therapy everywhere. At the moment, the ACTs are still effective, it seems, but good surveillance of those drugs, especially in those countries, is of course very, very important because we are very afraid that the same scenario as we have seen in the Greater Mekong sub-region, where you lose efficacy of the partner drugs, will also happen in these African countries. In addition, we think that also in the African countries, artemisinin resistance will translate to increased transmission of those parasites. Maybe the increase in malaria we currently see in Uganda is partly driven by this. By the way, that points to a possible intervention that we could do, which is deployment of single low-dose primaquine. Which is recommended as a gametocytocidal drug in low transmission areas because then you reduce transmission of malaria in general. Usually that is not recommended in high transmission settings because there you have that big parasite reservoir in asymptomatic patients. But if it preferentially inhibits the transmission of artemisinin-resistant infections, you could say, maybe we should deploy single low-dose primaquine independent of the transmission intensity. And I have a slide on that later. And of course, the resistant parasite can spread to neighboring countries. If you look at the trajectory of artemisinin resistance over the last few years, it has been modeled that the problem will become worse and worse over the coming years, and we need to intervene before it reaches those very high prevalence levels. So what can we do? Well, non-artemisinin-based combination therapies, that of course would be a solution. And there is quite a diverse antimalarial pipeline being developed at the moment, but nevertheless it will take at least a couple of years before those drugs will become available. There are a few front runners, especially the current Novartis drugs. So cipargamin as a drug to treat severe malaria, ganaplacide and imidazole-piperazine to treat uncomplicated malaria, where it is being developed in combination with lumefantrine. Those are furthest in their development, but nevertheless still years away from development. And we have seen over the last years that there can be very good candidates that still don't make it to an end product along the line of their development. Many other drugs under development even further away from being marketed. So at the moment, we have to work with the drugs that we have available now. So what can we do? I listed here the potential things that we can do. A thing that our research unit is heavily investigating at the moment are so-called triple artemisinin-based combination therapies, where we combine the artemisinin component not with a single partner drug, but with 2 partner drugs, as we, by the way, also do in the treatment of HIV or tuberculosis. And these are the combinations that are currently being tested. Artemether lumefantrine plus amodiaquine. I have some more information on that. Artesunate mefloquine plus piperacin. Arterolane piperacin plus mefloquine. And artesunate pyronaridine, atevocrone, proguanil. I'll tell a bit more about that, but another approach is multiple first-line treatments where you do not put 2 different partner drugs in a drug for a single patient, but in an area you deploy several artemisinin-based combination therapies next to each other, so that the parasite population sees different drugs when they jump from one patient to another. That has been around as a concept for quite a long time, and if the effects are being modeled, yeah, it reduces antimalarial drug resistance, but it has not been deployed on a wide scale in any country, even not in the Greater Mekong subregion. Other approaches are increase the duration of the therapy, for instance, of artemether lumefantrine from 3 days to 5 days, or sequentially treat a patient with 1 ACT followed by another ACT. So what of these possible strategies are the most effective? By the way, I put here a picture of the only artemisinin-based triple combination therapy that is now being developed as a fixed dose combination by Fosun Pharma, which is artemether lumefantrine amodiaquine in a single tablet, which is furthest in its development as a triple ACT. So which of these 4 strategies are the most effective? Well, that's very difficult to test in a clinical trial, so we have to rely on modeling efforts, and there are several groups who have done that. Maciek Boni has developed the model that's shown here. Our own unit, together with Oxford University, has developed a similar model. They both point into the same direction, that deployment of the triple ACT combination therapies is a very effective way to stop the spread of artemisinin resistance. And they perform better than the multiple first-line treatments or prolongation of the duration of therapy. So this might be a way to go forward. Why do triple ACTs stop the spread of multiple drug-resistant parasites? Well, that is because if you combine the 2 partner drugs, which have a bit matching pharmacokinetic properties, then after a 3-day therapy, where the artemisinin component will have disappeared rapidly because it has a plasma half-life of only 1 hour, the remaining parasites see 2 different partner drugs. So if the parasite becomes resistant to one of the partner drugs, it will still be killed by the other partner drug. That is simply speaking the rationale and the mechanism why it is good to give 2 partner drugs when you combine it with the rapidly eliminated artemisinin component. And our research units have done several trials now testing these triple artemisinin-based combination therapies, mainly artemether lumefantrine plus amodiaquine and DHA piperacin or DHA piperacin mefloquine or artesunate mefloquine plus piperacin. 2 big studies. First was the so-called TRACK-2 trial, here in countries in the Greater Mekong subregion and in India and Bangladesh. more than 1,000 patients enrolled, and we could show that the triple combination is very effective, also in those places where the ACTs were failing, and they were quite well tolerated and safe to use. We saw a bit more vomiting, especially when we added amodiaquine to artemethyl-lumefantrine, but it was much less the vomiting rates than compared to artesunate amodiaquine, which is widely used in Africa. And that is because we split the dose of the amodiaquine to match it with the twice daily dosing of artemethyl-lumefantrine. Another side effect that we saw was bradycardia, also attributed to amodiaquine, but which we don't think has important clinical consequences. Another study we did was comparing artemethyl-lumefantrine plus amodiaquine versus AL, artemethyl-lumefantrine, in Cambodia, where we have very widespread artemisinin resistance. Well, it performed, efficacy was a little bit better than AL alone, but it was quite a small study. This is now supplemented with the DTEC trial, which is a big trial, again evaluating triple ACTs, artemethyl-lumefantrine and amodiaquine, as well as artesunate mefloquine plus piperoquine, in both Asian but also in many African countries. The results of this are almost there. So recruitment has now been completed. The results have to be analyzed and will be communicated with you sometime soon. But just a little bit too early to show you these very exciting results. But just I cannot stop myself by showing that also here artemethyl-lumefantrine plus amodiaquine was better than artemethyl-lumefantrine in Cambodia. Here you see that. If you combine the three trials we did, so the TRACK-2 trial, the TECT-CV trial, I showed you the results, and now the DTEC trial, it clearly favors the triple therapy artemethyl-lumefantrine plus amodiaquine compared to artemethyl-lumefantrine alone. I'm almost there. So I showed you before that another problem with artemisinin-resistant Plasmodium falciparum infection is that they seem to be higher transmissible with higher gametocyte rates. So if we want to deploy single low-dose primaquine at a higher scale in African countries, we have to show that it is really safe, because primaquine is an oxidative drug and can cause hemolysis in G6PD deficient individuals. So that had to be investigated in African countries. So this was done recently and published in Lancet Infectious Diseases, where it was shown in sites in Uganda and the DRC that single low-dose primaquine, as expected, rapidly reduced gametocyte prevalence over time, but that it was also very safe to give even in patients that were carrying the A- allele for G6PD deficiency. So it did not cause severe malaria, severe anemia in this group, which would, I think, we are now at a decision point. Do we say we have to deploy now single low-dose primaquine on a much larger scale in Eastern African countries, or do we need further proof to show that you preferentially reduces artemisinin resistance falciparum malaria infection? Because you know, a lot of the transmission is also by the untreated patients in the asymptomatic reservoir. And then there are the non-drug treatments that need to be widely deployed, which are also important for the African context. where artemisinin resistance now has appeared. The malaria vaccine probably has to be rolled out wider. As you know, in addition to RTS-A01, we now have the R21 matrix M vaccine. Spatial repellents is another intervention that can be tried. Ivermectin as an endectocide, killing the parasite in the mosquito, probably has to be deployed wider. In addition, it's important to look at the health system in the Greater Mekong sub-region, which has been hugely successful, is the setup of the village malaria worker networks for early diagnosis and treatment, for other interventions, for surveillance. That's, of course, community health worker networks also exist in many African countries, but could be clearly developed further. Also, the collaboration between NMCPs, the National Malaria Control Programs, and all the other stakeholders, including civil society organizations, NGOs, academic groups. We all have to work together, and we have to work together with private companies to develop new drugs and new approaches. We are in here all together. And of course, WHO should play an organizing role there too. So this is my last slide. This was a talk about the spreading and increasing problem of artemisinin partial resistance. The artemisinin resistant phenotype shows itself in delayed parasite clearance in patients. It is because of reduced ring stage susceptibility towards the drug, but also mature Schison state susceptibility. It increases transmissibility because the gametocytes also become less susceptible to the drug. And intrinsically, it seems that these artemisinin-resistant infections are more transmissible. It facilitates the development of partner drug resistance, and then we are in real trouble because then the ACTs become less effective. The whole situation is in flux. It's emerging now in Eastern Africa. It's a huge concern. It will be a big topic at the MIM conference that starts next week in Kigali in Rwanda. What to do about it? Here in the GMS, we have to eliminate falciparum malaria. We have big worries about Myanmar because of the political situation. For the African situation, triple combination therapies might be a way forward. Wider deployment of single low-dose primaquine improves surveillance, both molecular surveillance and TES studies, whether the ACTs still work. Better antimalarial stewardship. I didn't mention that, but there is probably an overuse of parenteral artesunate monotherapy in patients with uncomplicated falciparum malaria and other tools. New drugs, very important, but will take a few more years. Thank you very much for your attention.
Thank you so much, Arjen. That was quite an elaborate discourse in looking at the journey with respect to anti-malaria medicines and the evolutions around anti-malaria resistance and the various thoughts that we have. We have not had too many questions, but I would like to ask us to please use the Q&A. We are slightly over the hour, but we will spend about another 5 to 7 minutes thereabout for the Q&A. So the first question I have here, Arjen, is that what do you think about the role of malaria vaccine in the containment of malaria resistance? I know you were mentioning that towards the end, but maybe you have more time now.
Well, non-artemisinin, non-drug interventions, I think, are very important because all the drugs we have contain the artemisinin at the moment, so that increases drug pressure, whereas the vaccine is a whole different angle to reduce the transmission of falciparum malaria. So I do think it is important. RTSS, as you know, is not that effective, but now we have R21 matrix M, which seems to be more effective. So I think that wider deployment of the vaccine is very important to also tackle the problem of drug resistance, but not on its own. It needs to be embedded in a very broad intervention package to tackle this problem.
Thank you very much. We have some other questions. So there's this question from Owen. It says, forgive me if I missed it. What is the proposed mechanism of reduced mature schizon susceptibility in resistant parasites? Is that also a K13 mediated or thought to be something else?
Yeah, that is a good question. So artemisinin resistance clearly in the young ring stage, but also in the mature schizon stage, when you look phenotypically at it in vitro culture. There's one publication that says it's related to increased rosetting of these mature parasites, which is conceptually not that evident that can be a reason why the schizon stage parasite becomes less susceptible to artemisinin. So the honest answer is that we do not really understand the biological mechanism clearly at the moment. But it is also closely linked to the CALC mutants.
I think we just have about 3 more questions, very quick ones. One here says, have any East African countries with artemisinin partial resistance expressed interest in rolling out triple ACTs?
Well, yeah, so I'm completely biased here because we are developing these triple ACTs. We have engaged with NMCPs and other groups in those countries. They are clearly very interested in the concepts. That said, they are not available yet in the market. So Fosun Pharma now makes the fixed-dose combination artemether lumefantrine amodiaquine. We will start a trial testing these next month, but then it still will take 2 years before they really become available. So there is clearly interest, but it will take some time before they are ready to go to being deployed.
All right. So now we take just this other 2 questions. It says, in current context of WHO recommendation for trigger for therapeutic policy change when threshold of sensitivity is below 90%, and in many countries following these guidelines will not want to proactively switch their ACTs where their TES show good efficacy. So what will be your message for this? That is readiness where they still think that their TES result still shows efficacy significantly above 90%. Do you see them having a buy-in into triple ACTs? And you may want to share your Nigerian experience.
Yeah, that is a very relevant question, of course. So good surveillance, of course, is important for that you really know that the efficacy of your drugs stay above 90%. You could make a case that you deploy the triple ACTs before you start failing of the ACTs. because you prevent the development of artemisinin and partner drug resistance. But you can only recommend that if it is affordable and not at the expense of more side effects. So that needs quite a bit of discussion before you can make a hard recommendation for triple ACTs before the arrival of artemisinin resistance in an area. But if you want to be on the safe side, I would still recommend that.
Yeah. And probably that brings us to the whole concept of stakeholder engagement, where we already have some evidence that people will have a preference for it if shown to have some slight edge over what they are doing currently. Now to the last question, it says, with reduced host immunity against falciparum malaria, how might that affect the spread of artesunate resistance?
Yeah, that is a good point. So very much in general terms, partial immunity or premunition will reduce the spread of drug resistance because parasites that have reduced sensitivity but are not fully resistant, they are helped by the host immunity to clear the infection. So if you are losing that, you expect increased spread of drug resistance, including artemisinin resistance.
Okay. Finally, I think Tama Ghosh would like to ask a question live rather than from the Q&A. So I'll bring her in.
Thank you, Professor. So no question for me, actually, but an enormous thank you. Thank you, Professor Dondorp, for your talk. I have to say we were really pleased in the team, obviously a long-standing friend of the Society. We were delighted with your nomination and award. And so also thank you, Professor Mokwolo, for nominating him. Thank you both for spending the time today, sharing an important area of research with the community. For others that have been listening and will be listening a bit later, there are lots of medals and awards that we give out to recognise success like this. There is a deadline tomorrow for 3 of them, including the Emerging Leaders Award, Chalmers Medal, and the Donald Mackay Medal. So all at different levels of career and seniority and success. But do have a look at those and into the future if you have an interest in recognising yourself or a fellow member of the Society. Thank you again, Professor Dondorp and Professor Mokwolo, for your time. Thank you so much. And to everyone, we look forward to seeing you again another time. Thank you and enjoy the rest of your day.
Thank you very much. Bye.
Thank you.
Thank you. Bye-bye.